So good afternoon. First, I'd just like to thank Claire for inviting me to come and talk about my research. I think one of the things which is really nice about the talk this morning is the constant reference to the patient. And I think it's kind of unusual for science. We tend to fall in love with the technology and forget about the end user and the person who's going to be impacted by the technology. And that kind of comes to, I suppose, the way we structure our research. So about 10 years ago, we got funding for medical technology. And one of the things that I was really passionate about was we've just got this funding. We need to have a patient impact. So what we wanted to do over a 4-year period was get our device into the clinic. So one of the first things we did was we looked nationally and internationally at groups who were developing technology in our space. And at that stage, there was 180 researchers developing technology or developing medical devices using the same type of technology. And out of that 180, only 10 had ever tested their device on a single patient, and only 1 had ever sought to commercialize the technology. So this to me is extremely scary. So we had been given investment to deliver a new diagnostic at the time. And how are we to do that in an area where it's so hard to actually get the technology out of the lab? And one of the things that we saw was that, medtech, developing a medtech company or commercializing a medtech device, it's like a normal startup, but just with a lot more ways to fail. So about 75% of all new medtech companies fail within the first 5 years. And the average cost of getting a medical device to patient is about $9 million. So as a kind of scientist, we put our science hats on and we said, okay, so let's just see how the 10 of our 180 who've actually done it, what's common amongst them. And there was kind of 2 things that became very clear. The first is that the ones that were getting their device to the clinic had strong clinical partners. And so not just collaborating, but almost being co-located. And the second thing was that they were thinking about that funding pathway. So the second part here at the very start. So how do you fund this? How do you not just develop technology and let it stop within the lab? So what we've done is essentially tried to replicate that here in Galway. So we've essentially moved our entire engineering lab into the hospital. So now we have a group of about 23 engineers who are working on medical device labs or projects in the hospital. We're co-located with the clinical trials facility. And it's this idea of trying to incorporate the voice of the patient, of the clinician, and of the investor as well. So how do we show to an investor that this is technology which will address a real clinical need and should be supported? So we have a group, interdisciplinary group. So we have 23 engineers, clinicians, physicists, veterinary surgeons, all working within the hospital, developing new diagnostics and therapeutics. And my own desk actually sits in the middle of the lab. And one of the things that happens a lot is that the clinicians are passing, and sometimes they'll say, I have this problem, and maybe you'll have a solution for this, or maybe we can brainstorm a solution for this. And one really nice project that came to me from an endocrinologist, Conal Denedy. So I'm going to mention it because this is his project, this is his baby, and we're just helping on the technology side. But he was talking about Con syndrome, which I'd never heard of before. But essentially, 18% of people who have hypertension, it's caused by this syndrome called Con syndrome. And these are very small hormone-releasing tumors that grow on the adrenal gland. And he presented this to me, and then he said, so the problem is we just have to get rid of these benign tumors, and if we did, we'd cure hypertension in about 20% of the population. And when I saw that, I was like, this is crazy. Why isn't somebody developing this solution? Because hypertension as a disease is so complex to treat, there's so many factors, but here's 20% of the population which you could treat by just getting rid of these small tumors. So that's the next question I asked him. So how is it treated now, and why isn't this being fixed? And this is an example of the tumor growing on the adrenal gland. The first treatments they do is adrenalectomy, which is you remove one of the adrenal glands which has these tumours. And the risk there with that treatment, and this often happens, is that the tumours will start growing on the other gland. And then your next solution after that is essentially drug treatment. So these are complex drugs, they have lots of side effects, and then a lot of the time the patient, because of the side effects, chooses not to take them. So it's not managed very well. And the last solution, which was kind of the first solution that came to mind for me, was radiofrequency ablation. So this is a radiofrequency ablation device. And essentially what a radiofrequency ablation device is, just a hot metal tip that's introduced into the tumour. It heats up and essentially kills the tumour cells, and it's used for treating tumours in the liver and tumours in the kidney. But the problem with previous studies that have used radiofrequency ablation is they've essentially cooked the entire adrenal gland. So it's a very, as someone described to me, it's like using a sledgehammer to crack a nut. It's just way too much power and very imprecise. So again, the next question I asked him is, what does an ideal solution look like? And this to me is really interesting. So the key problem is that you want to protect healthy tissue first. So all of this tissue on the edges is functional tissue, and you need to protect that first. And this is kind of the opposite way to the way radiofrequency ablation was designed. That was designed firstly to kill cancer tissue. So you want a really hot area, you want a large ablation zone, and you want to destroy as much tissue as possible. And what Conal was asking me for was a device which does the opposite. So it firstly protects the tissue, the functional healthy tissue, then you treat the tumour in the middle, and then at the end you will hopefully restore the normal function of the gland and just have this small ablation zone. So as I said, the problem with radiofrequency ablation is it produces this large ablation zone. It basically kills all the cells at the middle, and it produces this kind of teardrop shaped ablation zone. So it's basically the wrong tool for the problem. So we had some expertise in microwave ablation. So microwave ablation produces a very small ablation zone. Because of that, it protects the surrounding functional tissue, and by design it produces this very small circular shaped ablation zone. And this technology is not complex, it's actually quite straightforward, but it hasn't been used, I think, widely in clinical practice, because up to this we were trying to treat large tumours. These are very small tumours, and the goal is very different, which is protection of functional tissue. So just to show you what it looks like, on the left you have a radiofrequency ablation zone, which is this large, hot, tear-shaped zone. And on the right you have a smaller circular ablation zone, which we get from microwave ablation. So when we were brainstorming this, we were thinking, okay, now we can treat potentially treat the tumor, we can protect the functional tissue. How do we want to deliver it? And exactly as Niamh was saying, we want it to be minimally invasive. We want very low risk. We want this to become a simple procedure to perform. So we were looking for, this is kind of our concept, which is an endoscopic device, which is delivered via the stomach, a minimally invasive probe that protects the functional tissue, ablates the tumor, and removes the need for open surgery. So this was our pathway. Our goal is to get to patient in the same as all of our projects, is how do we do that? And really what we're trying to do is to gather evidence, constantly gather evidence. And the first step was, let's build a prototype and let's test it on ex vivo tissue. Then we move into animal tissue. And this is kind of the standard development pathway for any medical device. You look at short-term animal studies, and then you go to long-term animal studies, and you see the acute effect and also the long-term systematic effect. So this is now, the project is running for about maybe about 18 months. So this is our first generation probe. So we developed this microwave catheter, very small diameter, maybe 1.5 millimeters in diameter, and you get this. So we're essentially testing it on ex vivo tissue on the bench, and you get this very nice 10 millimeter diameter ablation zone. So I really like this project just because it's so simple. It's not complex science. We're essentially just heating tissue in a really controlled way. One of the challenges then is that we want to do this procedure quickly. So we want to deliver as much power as possible in the shortest time for the safest procedure for the patient. And if you deliver a lot of microwave power in a short period of time, essentially the entire cable starts to heat up. So our next generation prototype, we put in a cooling system. So we now have liquid cooling, which goes down this 1.5 millimeter bore and is constantly cooling the probe, so we can deliver a lot of power at the tip and very quickly treat that tumor. So I like this diagram just because I think it's kind of the kid in me. You can put these probes into egg white and you switch them on, and you can see in real time the ablation zone generating, because it's essentially cooking the egg white. But it's a really nice way to quickly test lots of different designs. So then we said, okay, let's move quickly to animal studies. So we just before Christmas we went to Kansas State University, who have expertise in large animal study, and we did a 2-arm study. So we had 7 animals, and approximately half the animals were treated with our device, and the other half remained untreated. And then we followed the animals for 1 week to see the acute and chronic effect. So we were ablating the adrenal gland in the animal and then seeing, okay, were we able to create a small ablation zone, and did they have normal adrenal function afterwards? And did they survive the operation? So all of the animals survived. All of them had their blood pressure went back to normal. There was no negative effect on the treated organ. So we were able, in our animal model, to develop this small ablation zone. The functional tissue was protected, and there was no systematic effect. I think one of the really exciting things that we saw in this study was that we tested various different amount of heat treatments, and what we found was even gentle heat treatment. So we didn't actually have to really heat the tissue. We saw it prompted the body's own immune system to kick in and started effectively killing the cells that had been pre-treated with heat. So this is a nice effect for making the procedure even safer. So we use smaller doses of heat over a shorter period. So this is where we are now. We're now moving towards our 3rd prototype. We will probably go to another animal study. But I think the key thing for the project is this idea of just connecting the engineering and the clinical team for impact. So we are constantly developing, on the engineering side, prototypes, modeling, managing and implementing the animal studies. And then in parallel, we have a clinician who's working with these patients on a weekly basis, giving us design inputs, telling us how this procedure needs to be, what our patients will look like, what the adrenals will look like, and also being able to do some of the science, so the histology and the treatment planning required to treat these patients. So just to really quickly summarize what we're doing, the impact of this project is huge in the sense that we have, hypertension affects about 30% of the population, and it's a major contributing factor to all of the diseases that we talked about today: cardiovascular disease, stroke. And this figure, every time I see it, I kind of think it looks just bizarre that 42% of deaths in Europe, this is a WHO figure, that hypertension is a risk factor. So this, to me, is just the impact of this type of work and the work that we saw in the earlier presentations is kind of staggering. Why I really like this project is we're just treating these small little tumors on the adrenal gland, and if we can do that, we can have huge impact on this 20% of the patients who have Conn syndrome. So it has many advantages. One is that it's drug-free, so it's a drug-free permanent solution. It's minimally invasive technology, and our goal is just to keep pushing it towards the clinic as hard as possible. I think that the biggest challenge for us is funding that first-in-man study, but at the moment our goal is just build as much clinical, technical and commercial evidence as possible to secure funding to do that. Thanks.
So if 18% of hypertensive cases have Conn's syndrome, that predisposes you to have to screen patients for Conn's syndrome first. So how is that managed currently? Is that through, elevated tumour markers in the blood, or patients, how is that, how are you going to access your patient population?
It's a really good point. And so we spent last summer in the States essentially talking to blood pressure specialists and asking them, are they screening for Conn's syndrome? So one of the difficulties with this is creating kind of a non-invasive way to detect it. So at the moment they do venous sampling. So they're kind of sampling blood from the adrenal glands, and they're saying, is there elevated hormones on one side? So that's quite an invasive procedure. So one of the things that Connell is interested in is looking for ways to more easily screen for it.
Are there no imaging techniques to do that?
So there are, it's a really good question. So there's a group in Cambridge who've got a functional PET scan, and you can see they show up on that. But the challenge with that is getting that out to all the patients.
Okay. And may I ask, just in follow-up, why specifically microwave radiation is targeted, or are there other, photo possibilities through laser that wouldn't have such maybe a strong heating effect, or is that absolutely necessary? You said about a gentle heat. Is there a way of modulating? I imagine for a lot of patients, when they hear, microwave radiation or that, it's...
Yeah, that's a really good question. I suppose, so when we're kind of planning a project, we think about the clinical, commercial and technical. And the commercial is a big part of it. So how do we build a device that will be at some stage attractive for someone to acquire? Because within a university environment, we're not getting 9 million to bring this to patient. So there's been a push with a lot of large medtech companies. Medtronic has a microwave system. So in every hospital now, they have microwave equipment. So we could, and I always say to our students and to our postdocs, we're technology agnostic. So whatever works. But in this case, it was a commercial decision that microwave seems to be gaining market traction, and it fits the technical requirements. Okay. Thanks.
I'd just like to say thank you again to all of the researchers, the scientists that came in today and gave time to give those presentations. If you think of questions after the fact, you can get in touch with me. I think my contact details are on the info pack, so and I'll be able to direct them. But what we're going to do now is change tack a little bit. We are delighted to have the CEO of CRE here today, Neil Johnson, who's going to give you a little bit of a different perspective on the work that CRE does around patient education and for both cardiovascular and stroke. So thanks, Neil, for coming along. And